This research paper, titled sEEG-based Encoding for Sentence Retrieval, a contrastive learning approach to brain-language alignment, is authored by Yijun Liu from the Ming Hsieh Department of Electrical and Computer Engineering at the University of Southern California. The work has been accepted for a poster presentation at the CVPR 2025 workshop on multimodal foundation models. The paper explores how to interpret neural activity from the brain by aligning it with natural language, using a method called SSENSE. SSENSE is a contrastive learning framework that takes stereo-electroencephalography, or sEEG signals, which are invasive recordings from inside the brain, and projects them into the same embedding space as sentences from a pre-trained model like CLIP. This allows for retrieving sentences directly from brain activity, essentially bridging the gap between neuroscience and artificial intelligence. At its core, the challenge is to decode what's going on in someone's mind by analyzing their brain signals. The authors build on recent advances in foundation models, which are large AI systems trained on vast amounts of data to understand relationships between different types of information, like images and text. Models like CLIP have already shown how to align visual and language data, but here, the focus is on extending that to neural data, specifically high-resolution sEEG signals. The paper proposes SSENSE as a way to map sEEG recordings into the sentence embedding space of a frozen CLIP model. This means they don't change the CLIP model itself. Instead, they train a separate encoder just for the sEEG data. The goal is to enable zero-shot sentence retrieval, where the system can match brain activity to the most relevant sentence without needing additional training for each new task. To achieve this, they use spectral representations of sEEG signals. First, the raw sEEG data is preprocessed by converting it into spectrograms using a technique called the supralet transform. This transform helps analyze the frequency components of the brain signals over time, similar to how it's been used in other studies like BrainBird. For consistency, they zero-pad shorter sEEG segments to a fixed length of about 4 seconds, which corresponds to 8,200 time points. The sEEG encoder is a neural network designed to take these spectrogram inputs and output fixed-dimensional vectors. These vectors are aligned with the 512-dimensional space used by CLIP's sentence embeddings. Because spectrograms have a visual structure, like images, the authors adapt a modified ResNet architecture for this encoder. The training process uses a contrastive loss function called InfoNCE, which pulls together representations of matching sEEG signals and sentences while pushing apart those that don't match. In practice, this framework is applied to a dataset where sEEG recordings are time-aligned with spoken transcripts from a subject watching a naturalistic movie. This means the brain signals correspond directly to the sentences being heard or processed in real time. Despite having only data from a single subject, which limits the scale, the method shows promising results in sentence retrieval tasks. The evaluation demonstrates that general-purpose language representations from models like CLIP can act as effective priors for decoding neural data. In other words, these pre-trained models provide a strong foundation that helps make sense of brain activity without starting from scratch. This approach highlights the potential of contrastive learning for brain-language alignment. By embedding sEEG signals into a shared semantic space, it opens up new possibilities for integrating brain signals with broader AI systems. Applications could include cognitive neuroscience research, where scientists might better understand how the brain processes language, or in assistive technologies like brain-computer interfaces for communication. The paper's findings suggest that even with limited data, it's feasible to align invasive neural recordings with natural language descriptions. This could lead to advancements in areas like language understanding systems that incorporate real-time brain feedback, or even personalized AI assistants that respond to unspoken thoughts. Building on this, the authors note that their work provides initial evidence for the broader integration of brain signals into foundation models. For instance, in movie-watching scenarios, this could mean analyzing how different parts of the brain react to narrative elements, potentially improving content recommendation systems or therapeutic tools for neurological conditions. In terms of the technical details, the sEEG encoder processes stacked spectrograms from multiple electrodes, treating the data as a multi-channel input. The contrastive learning setup ensures that for each sEEG segment, The model learns to associate it most closely with the correct sentence embedding from the clip text encoder, which remains frozen to preserve its pre-trained knowledge. Overall, this method underscores the evolving intersection of neuroscience and AI, showing how multimodal foundation models can be adapted for neural decoding. The results from this single-subject study paved the way for larger-scale experiments and more refined applications in brain-language interfaces.